Hello everyone, welcome to Twilight. Problem is possibility, and this is Dr. Rajlakshmi, and I am here today to talk about our third part of tropical infections, that is scrub typhus. This disease is a zoonotic disease, transmits from the animals to humans. Basically, human is an accidental host for this bacteria. This has been caused by the rickettsial bacteria, that is Orientia susumushi. It's an obligate intracellular gram-negative bacteria, and the vector for this bacteria or for this disease is trombiculid mites. It usually or actually has to infect rodents, but instead of rodents, it will bite accidentally the human beings. The larval stage of mites will transmit the bacteria while feeding on the humans. These are the mites. It actually has to infect rodents or other animals. Sometimes it can cause the disease for the humans. Following the bite, the bacteria multiplies at the site of the inoculation, and it can cause both local and systemic disease. Severity of the disease depends upon the strain of the bacteria and some human related factors, some host related factors. For example, if patient has G6PD deficiency, the severity of the disease will be more, the prognosis will be poor. The principal target site for this infection for this bacteria are endothelial cells. Once the bacteria enters into the circulation, it will infect endothelial cells of all the organs of the body. And it has been isolated from the macrophages in macrophages of the liver and spleen in few post-mortem cases. By the end of the first week, the immune system will get activated, try to eliminate the bacteria. Both humoral and cell-mediated immunity participates to eliminate the bacteria, but this bacteria will downregulate the expression of the glycoprotein 96. This molecule has been expressed or has been present in the endoplasmic reticulum of the cells. It plays a major role in defense mechanism against the bacteria. It plays a central role in major histocompatibility complex mediated antigen presentation and functioning of the dendritic cells, antibody production, and cross priming of the immune cells. But because of suppression of this molecule by the rickettsial bacteria or scrub typhus causing bacteria, that is Orientia susumushi, so it will increase the severity of the disease, and it helps replication of the bacteria in the host body. Co-infection with HIV will decrease the load, viral load of the HIV, but the mechanism behind this phenomenon is not clear. Coming to clinical features, the incubation period ranges from 10 to 12 days. It can vary from 6 to 21 days. The severity ranges from mild fever or mild febrile illness to multi-organ failure and multi-organ dysfunction syndrome. Fever, GI disturbances, cough, malaise, myalgia, headache. These are all non-specific symptoms and signs. Sometimes abdominal pain will be there. Rhabdomyolysis can happen in case of the scrub typhus. Maculopapular rash starting from the trunk, and it can spread up to limbs. We can see this rash by the end of the first week, and regional lymphadenopathy, one of the common finding in case of the scrub typhus. These are all maculopapular lesions. Can see all over the body, mainly on the trunk, followed by the limbs. ESCHAR is one of the pathognomonic feature of this disease, and this has been reported from 7 to 80% of the scenarios. The common areas to develop ESCHAR are damp and moist skin, mainly in the groin, axilla, and waist. Of course, we can see any part of the body wherever the mite bitten. The eschar starts as a small papule and that enlarges and subsequently undergoes central necrosis to turn into black color. Central necrotic part, that is in black color, and this is peripheral red part, inflammatory part. These are all maculopapular rash. In the second week of illness, where the immune system has been activated, we can see multi-organ failure in few cases, in few patients. Multi-organ failure means it involves all the parts of the body, especially central nervous system. It can cause meningitis, meningoencephalitis, myelitis, And cranial nerve involvement, cranial nerve paralysis, especially it can cause trigeminal neuralgia, sixth nerve and eighth nerve paralysis, so it can result in deafness. Sometimes we can see uveitis, pan-uveitis, one of the eye sign in case of the scrub typhus. Coming to cardiovascular system, it can cause myocarditis, arrhythmias, congestive heart failure, and vasculitis. Sometimes, in case of the severe infections, it can even cause infective endocarditis. Renal involvement can cause acute kidney injury. Sometimes patient may require dialysis. Rhabdomyolysis also can cause acute kidney injury. GIT, it can cause hepatitis, direct hyperbilirubinemia, pancreatitis, and diarrhea. Respiratory ARDS and interstitial pneumonia. Coming to diagnostic part. To diagnose scrub typhus, we should have high level of clinical suspicion and we should take occupational history. If a farmer presents with all these symptoms and signs, we should rule out scrub typhus along with other tropical infections. If patient is have presented you with the fever, myalgia, headache with leukocytosis or leukopenia, thrombocytopenia with liver and kidney involvement, should always rule out malaria, dengue, leptospira, and scrub typhus. So these all four diseases, especially in the Indian setup, we need to rule out when our patient presents with all these symptoms and signs. Coming to diagnostic test, WEIL-FELIX test is the oldest technique. Now we are not using it. It is neither sensitive nor specific. It's basically based upon cross reactivity of certain proteus species, certain species of the proteus bacteria with antibodies of the scrub typhus or antibodies of the rickettsial bacteria. For scrub typhus, we use OXK species of the proteus. For typhus fever, we use OX19 of proteus bacteria. For spotted fever, we use OX2, OX19. False positive we can see in case of the typhoid, proteus infections, and smallpox. This is basically outdated. Better not to use this test. Other test, the indirect immunofluorescent antibody. It's basically gold standard test to diagnose scrub typhus. In this test, we use fluorescent labeled anti-human immunoglobulins to detect antibodies in the serum of the patient. The fluorescent labeled anti-human immunoglobulins binds to immobilized bacterial antigen that was laid on a slide. So it basically detects scrub typhus antigens with the help of fluorescence labeled antibodies. But the main drawback of this test is single test is not confirmatory. We need to repeat the second sample, and we should be able to demonstrate a fourfold increase in the antibody titers. So basically, it will not be useful in case of the acute scenario. It basically for retrospective diagnosis. Indirect immunoperoxidase, it's almost equal to immunofluorescent antibody label test. But instead of immunofluorescent labeling, in this test we use peroxidase labeling antibodies. The sensitivity specificity almost equal to immunofluorescence antibody test. Coming to ELISA, the most commonly used test and commonly available test. With the help of this test, we can detect IgM antibodies in the serum and is most preferred test. Because WEIL-FELIX is outdated and it's not at all useful. Even immunofluorescence test, peroxidase labeled antibody test, all these tests are for retrospective diagnosis. For the ELISA, it uses recombinant P56 KDA type of specific antigen of this rickettsial bacteria, Orientia susukumishi. So that combines with IgM antibodies produced against certain strains of the bacteria like Carp, Cato, Gillium, and TA716 in case of the acute infections. So in this test, we try to detect antibodies against certain strains of the Orientia susukumishi with the help of antigen. So in India, we use the cutoff value of 0.5 to detect, to consider it as a positive test. Other test, immunochromatographic test, it's basically rapid test and point of care test. The basic principle is the same as that of the ELISA. We use the same antigen against the same strains of the bacteria, and the sensitivity will be 74 to 90%. Specificity is very high, 86 to 99%. Coming to PCR, highly sensitive, highly specific. It detects antigen even during second week of the illness, and very much even the parasite or the organism load is very much less. It will be able to detect the antigen. But the problem with PCR is it's costly and availability is also limited. Culture is bit cumbersome because it takes many days to get a positive culture, and the availability is very much limited. To maintain a culture media, the lab should follow biosafety level 3 containment and requires much technical expertise. The samples are taken from blood, buffy coat, skin biopsies from the experimental mammals. But it's this culture technique only for research purpose. Coming to treatment, the treatment of choice is doxycycline 100 mg IV BD or per oral morning and evening twice daily for a duration of 7 to 10 days. But now in India, there are many cases reported to be doxycycline resistant, and many treatment failure have been reported with the doxycycline. The another effective alternative is azithromycin. This azithromycin can kill the bacteria which are there inside the immune cells, macrophages, and leukocytes. Another alternative is rifampicin, but rifampicin is always the second choice of the drug because of emergence of rifampicin resistance, considering India is also an endemic zone for tuberculosis. Prophylaxis, we can use doxycycline, 200 mg of doxycycline weekly for the prophylaxis, especially for the high risk groups who are traveling to an endemic zone. So that's it for today, and we will meet up soon with another series of the lectures. I will end up our tropical series of lectures on the tropical infections. Stay tuned, and we'll meet up soon. Bye bye.